please go to menti.com. The code is in the upper right-hand corner. 11, 15, 12, 5. And please make sure you engage, right? So I can see the count in the bottom. I expect that we should be somewhere in the 20s. So let's get that number up into the 20s, right? 22. Right. So let's start by looking at what is chromatin. That's where I would start with this. So what is chromatin? Yeah. Exactly. So uncondensed, unraveled, unboxed. Looks a bit like ramen noodles, right? Okay, so why would I leave my DNA uncondensed like that? What's the purpose of that? Ease of access. Good. Your voice is clear. I'm just like under the heat, so, or AC or whatever they feel like running today. All right, ease of access. Exactly, right? The same reason you guys leave your stuff about your apartment, right, is you want to be able to access it when you need it. Remember, right, and a key feature that's going to keep popping up for us, that DNA has genes on them. And genes code for stuff, right? Important information that we need to get at on a regular basis. So in order to do our job, we need to be able to get at this information. My handwriting is absolutely atrocious. Right, and so chromatin allows us to do that. What do we call the boxed form then? Chromosomes. Good. I feel a little bit like the teacher from Ferris Bueller's Day Off up here. So remember, when we go to move our DNA around, we put it in this chromosomes form, which boxes our DNA up, it ravels it, and keeps it protected, just like when you guys move. All right, that's going to give us the information that we need, yes, thank you, in order to proceed forward and think about what is the best answer for our job here. Okay, so let's go through least popular to most popular. All right, least popular answer, metaphase. Okay, so we want to think about, are we using chromosomes here? Okay, so chromosomes are going to be present in mitosis, right? Because that's during cell division. Mitosis, growth and repair. This is cell division when we're moving things around. So, is my metaphase, that's a hard one, is metaphase happening during mitosis? Yes, right? So what exactly is happening in metaphase? We're lining up. What's lining up? Chromosomes. Chromosomes is like my adult banana, right? When you're a kid, you always do like the B-A-N-A-N-A-N-A thing. I do this with chromosomes where I want to like C-R-O-M-O-M-O-M-S-O-S-O-S-O-S-O. Like I just want to keep going. Check myself here. All right, so we have chromosomes in metaphase because it's part of mitosis. That's no good. I guess we'll just keep going since these were all like equally. All right, so anaphase is also not a terribly popular answer. Anaphase part of mitosis? It is. Okay. So what's happening in anaphase? Chromosomes are pulling apart. Okay, so what were sister chromatids are now chromosomes are getting pulled apart. Good. Okay. Now let's take a peek at telophase. Is that part of mitosis? Yeah, right? What's happening in telophase? Again? New cells are starting to form, right? But starting to is key here, right? So we still have 2 copies of our chromosomes in 1 cell. Okay, so that of course does leave us with your most popular answer, which is good, but we still want to check it, right? So what exactly is going on in interphase? Is this part of mitosis? What's happening here? Are cells dividing here? No. Okay, so we're not technically part of mitosis, right? Because mitosis is cell division. So what is happening in interphase? Okay, so we have cell growth. What else are we doing? Let's put it over here. I'm out of space. Okay, so we're making proteins and other things, right? In other words, we're functioning. We're doing our job. Work. Okay, so these 2 pieces are both really important to what's going on here, right? So our cell is living its best cell life. Okay, and if it has to be doing its job and growing in both parts, the cell and the DNA, I just lumped that together, we have to be able to get have the DNA to do that, right? I can't do my job, okay? I don't have the information to do my job if it's all bundled up and boxed. Okay? So my DNA would have to be in chromatin form for this to work, which means that you guys got this right. Yay! All right, let's do another one. This should have auto switched over for you on your screens, assuming you left Menti up. I'm waiting on 3 more of you. Right. Let's start and work our way to the top. What, if not prophase, is option 1? Okay. Telophase. Good. So remember when we're looking at these, this is a lot like what we've been doing in lab. Okay, the stuff inside that's very darkly stained is our DNA. So we're really focused on what is the organization of that really darkly staining stuff, right? That's our goal here. The sort of like dark outline is our cell membrane, or cell wall is appropriate. But if it's a boxed plant, so it's a cell wall. If it's a circle, it's going to be a cell membrane. That's, an animal cell. And all this light stuff that's not staining very well at all, that's going to be most of the rest of your cell material. Okay? That's not going to pick up the dye because, well, we don't want it to. So if we look at our number one, which we've identified as telophase, right, we have two distinct areas of darkly staining material. Right, so we have two pockets of DNA in one cell wall, but we can clearly see the beginning of a delineation in the middle, right? The beginning of cytokinesis. Right, so these are all hallmarks of our telophase. All right, great job. Let's take a peek at option 3. Anaphase, good. I'm old, you gotta give me a shot here. Anaphase. How do we know this is anaphase? They're separating or coming apart, right? So here again, even though it looks a little different, we want to focus on those dark bits, right? I have dark bits over here, dark bits over here. I have a very clear space in the middle, right? Right, and I can even look very dramatically like they have, right, a tug, a direction to them. Okay, so that's my anaphase. So you can see that space. Very good. All right. So now let's look at our final two options. This is usually the hardest. So let's look at option 4. What is option 4? Interphase is correct. And this is a little bit of a tougher one to look at. How did we know this was interphase? We still have a dark part, but what's different about it? So everything that's in that dark part is still our DNA. Right, so the DNA is uncondensed, right? And that makes it look different. So let's compare this to our correct answer, right? This is the only one left, so it has to be prophase. Let's take a peek at why it's correct. So how did we know this was prophase? So we can see the chromosomes, right? Okay, so we see clearly condensed chromosomes. Wow, okay, spell that. Are they organized in any special way? No, right? And that's important because in prophase, that's our key, right? We haven't done anything with them but boxed them up. Right, so that's how we knew. And this is one of the ways that we can really tell prophase and interphase apart. Right, is in prophase we get these clearly condensed pieces. When we look at interphase, oops, back to my wrong answer color. It doesn't really look like anything. Okay? And like someone mentioned, that's because everything is unraveled here. Okay, so we don't see those clearly condensed chromosomes because there aren't any. Okay? Like Simone mentioned, you can still see the nucleus with the DNA in it. That whole area is still darker than it should be, right? Because the nucleus does still have DNA, but the DNA isn't obvious, right? They're not condensed into very clear pieces because it's the chromatin form. So it's just like blah, blah. It looks very fuzzy. All right, great job. That's a tough one. I should help you with your lab practical. All right. You're recording, yes? I don't even know why I worry. All right, so we had finished up mitosis before. So this review is both helpful for what we're going to talk about next, and it'll help you for your lab as well. That's a really good way to study stuff for both. Okay, but the next thing we're going to talk about is control of the cell cycle. So now that we understand the stages really well, the last thing we want to talk about for mitosis is how do we make sure it works? So the first half of this is really going to be looking at checkpoints. And how do we make sure that all of these things are proceeding as they're supposed to? And then the very last thing we're going to look at is what happens when things go wrong. Okay, so what happens when some of these checkpoints or these control factors fall apart? And how does that create some of the things that we're sort of accustomed to hearing about, right? Like tumors and cancer. Okay, so some of the major checkpoints that we're going to look at. G1 and G2, which we'll look at sort of collectively, right? So our growth checkpoints. Right, so these you can imagine are important. We want to make sure before we move on to either of the next stages, the cell needs to be big enough, right? We don't want to proceed to any replication stage with a teeny weeny cell. Okay, so then it doesn't have enough cytoplasm or enough mitochondria, whatever. Okay? Certainly we can imagine would be extraordinarily problematic. So we need some kind of checkpoint to make sure, are you big enough, right? You must be this tall to ride the ride. Okay. The other checkpoint we're going to look at is the M checkpoint, right? Meaning the metaphase checkpoint. And there are more checkpoints in this. We're going to look at this one in particular because, as we sort of alluded to before, it's extraordinarily important to literally have all your ducks in a row. I'm on game today. Okay. So, right, we want to imagine having everybody in metaphase lined up. Okay, so the idea is that as we split in half, everybody gets exactly the same, right? Equal shares of the pie. So if we don't have everybody lined up there, okay, you could potentially end up with one half of the cell with more chromosomes than the other. So then the final result would be not sameness, right? So we would not have achieved our goal, and that's hugely problematic. Okay, let's take a peek of this. Starting and thinking about growth checkpoints. Excuse me. The two major pieces we need for these checkpoints are called cyclins, cyclin-dependent kinases. Okay. Both are made out of our favorite biomolecule, proteins. Okay, so let's start by talking about cyclins. Okay, so cyclins, okay, are produced as the cell grows. Okay, so that's what we mean by in synchrony. So along with. Okay, so as the cell grows up, right, it's making more cytoplasm, more plasma membrane, blah, blah, okay. As it's doing that, it's also making cyclins. It's like a side project. Okay? These are going to serve as keys, okay, to help them pass through the door into the next checkpoint. So we're making keys. That's the way I like to think about them. Okay, these keys are going to go into a very specific lock. Okay, that lock is an enzyme. Okay, and like all enzymes or all locks, okay, they're very specific. Remember, this is one of the things we learned about enzymes, right? Is enzymes are very specific to their substrates. They're very specific to their reactions. Okay, so this enzyme is very specific. Okay, it wants cyclin as its key. Okay. Plus side, despite the fact that its name is unwieldy as the daylight, it at least tells you what it wants, right? Cyclin-dependent. And then the kinase part, remember this ACE ending we learned in lab tells us it's an enzyme. Cyclin-dependent kinase. I want cyclin, I'm an enzyme. Okay, so this is the lock. Okay, the locks already exist, right? Just like any old house or any old door, the lock's already there, but you can always go make keys. Okay, these two pieces make sense? These are the two pieces we need for our story. All right, here's how this works. If we look at one of our growth checkpoints, okay, so let's look at the G1 checkpoint. I'm a bebe cell, little bebe cell. And I want to get into the S checkpoint where I can synthesize or duplicate my DNA, right? So step one, okay, I'm growing, I'm making more cytoplasm, I'm making more plasma membrane, I'm becoming a big adult cell from a bebe cell. And while I do that, I also make keys, aka the cyclins, right? And so as I get bigger, I get more cyclins. This is happening at the same time. The bigger I am, the more keys I have. That's not so bad, right? Step two. My keys go into my locks. Okay. So those cyclins are going to bind with the cyclin-dependent kinases, the enzymes. Okay, so my cyclin proteins bind with my kinase enzymes. Keys go into locks. But this only works if you have enough keys. There are many locks. There's more than one. One of those big mansion type houses. Okay, that makes sense? Okay, so this is why it's so important that we've been making keys the whole time we've been growing. We need to have made enough keys to fill all of the locks. Okay, so then at step three, and we've unlocked all of the locks. So we can now trigger S phase. Right, so we can open the door. into S phase. Okay, so the details here tell us that the enzyme phosphorylates. Okay, this means I add a phosphorus to some molecules that unlock a door, right, that allow us to get into DNA replication. Okay. I will never ask you about these kinds of details, but a lot of you always ask me about some interesting facts, so we have those. But the key here is once we've managed to bind enough cyclins to kinases, okay, we can now trigger into S phase. Okay. So this whole process is held back by that growth phase. If I haven't grown enough, if I haven't made enough cyclins, if I haven't made enough keys, I won't be able to pass into S phase yet because the door won't unlock yet. So this is what we mean by a checkpoint, right? I'm held here until I've managed to accomplish this task. Okay, any questions about our cyclin and kinase key and lock checkpoint? Okay, let's look at another type of checkpoint. This is at the spindle checkpoint. On our visual diagram, we call this the M checkpoint. We're at metaphase. Okay, so we're stopped at metaphase, okay, with the goal to get into anaphase. Okay. So we want to get into anaphase, and this is done through a third body called the anaphase promoting complex. Again, on the benefit here, something that tells you what it does. Okay, so the key here is, while we were sort of imagining our movers, right, just pulling and pulling and pulling on their spindle fibers, and the kitkats come apart. Okay? Luckily, that's not actually what happens. Okay? So this APC, our anaphase promoting complex, is going to come through and actually check, right, that everything is lined up, all the tensions are right, everybody's connected, and that's key. Okay? Up to this point, the movers can pull on their spindle fibers as hard as they want. Okay? Our sister chromatids are not going to pull apart. Okay? They're stuck there. They're glued together. Okay? What we need is the say-so from the boss. Okay? And this anaphase promoting complex is the boss, and he has to sign off before anything can happen. So when they do, a signal comes through the cell that activates a set of proteins that removes the cohesion. So the cohesion is like the sticky stuff. At those centromeres that's holding our sister chromatids together. And then, as our movers pull on the spindle fibers, our sister chromatids will come apart. Okay. So it's like dissolving the glue in between them. Kind of like when you have to use nail polish remover to get super glue off. And you need somebody to actually do that. And the APC is doing that. So this is good, right? It's making sure that nobody gets jumping ahead, or someone is actually making sure everybody is where they're supposed to be, right? Quite literally, all my ducks are in a row before we spread them apart. Did that end up answering your question? Any questions about our metaphase? Ooh, I cannot talk today, guys. Our metaphase checkpoint. Okay, let's broaden our outlook then at how cell division is controlled. So the two checkpoints we looked at are very specific, right? The cyclin kinase business work with our growth checkpoint. Okay, the APC specifically is an anaphase promoting complex. It's only going to work with anaphase. But when we're talking about things like tumors and cancers, these are umbrella terms. Okay? That's why they're so hard to treat and to catch, right? Because it really is just talking about uncontrolled cell growth. Okay, so cells that shouldn't be growing, okay, or in parts of the body where they shouldn't be. And that's a big umbrella thing. So two of the most common types or classes of genes are responsible for when this goes down. Now I say classes, so there's very specific genes in these subsets where this occurs. Now these genes are good genes. So we're going to talk about them. These have jobs that help. Right? They have good jobs. So when we have things like cancers or tumors occur, they busted. Okay, so something has went horrifically wrong. So the two types of classes we're going to talk about are tumor suppressor genes. Sounds like it's doing something, right? Okay. And then proto-oncogenes. Okay, so pre or preventative of oncology, right? Cancer. We're going to talk about what these do or what they're supposed to do, and then what happens when they, quite frankly, stop doing it. So let's start with our tumor suppressor genes. This is a whole class of genes. Okay, so there's lots of different genes in the body that perform similar type functions. Okay? Their overall goal, a mighty goal, okay, is to prevent the development of cells and cell lines that have mistakes in them. Okay, so this is a big deal and a big job. So I akin these to sort of like the traffic cop in the road, okay, who's stopping drunk drivers, right? So like the drunk driving check, okay? So he's not at the party making sure that people aren't drinking too much. He's not at the bar, okay, keeping people from drinking. There's not any of these checkpoints that we've talked about, right? So he's not stopping cells from getting damaged. He's the next point of defense to make sure that that driver isn't continuing down the road and damaging anybody else. And that's the key here. So we're going to go through a very specific example, a really common one, the P53 gene. And I say common as in like people kind of know about it. I've seen memes about it. And I know I follow dorky meme boards, but like they exist, so that's something, right? Okay, so this is going to halt cell division when damaged DNA is detected. Good thing, right? If this is absent, if this gene is absent, or if it's damaged, so it's not made accurately, okay, this is where a lot of cancer cells come from. So let's walk through an example. In my top or my blue box is how our traffic cop is supposed to work, right? So here I have some damaged DNA. Okay, this might have gotten messed up in mitosis. Okay, more commonly, perhaps, we had maybe some exposure to some sunlight. Okay, so we damaged our DNA. Okay, so we've detected the damaged DNA. Our traffic cop, right, our P53 gene, makes a P53 protein. It's our favorite biomolecule. Okay, comes in. Okay, he scans the DNA, and catches the mistake, the damage, right? Okay, cell division comes to a halt. Okay, we catch this line, stop, stop. Okay. Now, there's two things that can happen when we find out that DNA is busted up. Okay? It depends on how bad it's busted up. Okay? If it's not a super big deal, and we'll end up talking about this when we talk about DNA replication, there are enzymes that can come through and fix the problem. Okay? Remember, we have blueprints, so we can come through, okay, send in our guy. Okay? He or she can come in, okay, find the damaged pieces, use the blueprint, and put in the right parts. Okay, so we can fix it. Edit, repair. Okay, if we do that, then the DNA can go on and live its merry way. Okay, that cell can go forward, live long and prosper. Not so big of a deal. So it sits in the drunk tank for a night. Sometimes, though, the DNA damage is bad. Okay? Unfixable. The enzyme goes in, has no idea what's going on, there's breaks everywhere. I can't fix this, man. You're getting notes from someone and you're like, what does this say? I don't even know where this is. Did they write anything down for today? Okay? That kind of bad. Okay? So if that happens, our P53 protein has to take drastic measures. This is where my analogy stops, right? So if that happens, we have to do what's called apoptosis. Okay? We can't let this cell line continue. We don't want a whole bunch of cells made out of damaged goods that don't know how to make livers, okay? Don't know how to pump a heart. You cannot continue. Okay, so stop the cell. If there's anything good, we recycle that for spare parts, and otherwise, okay, we dissolve it into trash. We fill it with enzymes and let it die. Let it go. Let it go. Right, so that's how it's supposed to work. I'm a little hyped up today. So here's the problem. So let's focus on my green box. If our COP is missing, okay, so we're missing the P53 gene, or our P53 protein was made improperly, right? Now I still have, or I'm starting with my DNA damaged cells. So I still went out in the sun, right, ruined my DNA and some cells. So I don't have anyone to check those cells. So they are living their best cell life. And that's a huge problem. Because remember, in mitosis, I start with one cell, right? One cell becomes two cells. Try to get two daughter cells off of that. Each of those two daughter cells makes two daughter cells. So on and so forth. So now I have, first off, a whole bunch of cells that have that one defect. Okay? And the likelihood that a defective cell doesn't continue to make more defects. Right, so this is where the problem therein lies, right? Now we've got a bunch of busted up cells living their best life, causing a ruckus. This is suddenly a pretty bad neighborhood. Okay. And this starts out, right, all in one pocket, right, one part of the tissue. Going and going and going and going and going. They don't know what they're doing. They don't know how to function. They're just going and going and going. With me? Feel good? Okay. The other type of gene, remember, or the other type of class, are called Proto-Oncogenes. Okay, so remember onco like oncology, cancer. But proto meaning before or preventative. So again, these are normally good genes. I like to call these the mom genes. These are like extreme mom genes. Get it right, mom genes? These are like helicopter moms. They're called snowplow moms now or something. Right, so they normally are in charge of every waking minute of the cell. Okay? Top box is telling you, okay, I tell the cell when they should be growing and when they should be not. Right? How big should you get? And you should be growing now. Okay? You're just a growing boy. Okay? My second box is telling them when to work. Right? Making proteins, when to communicate, that's a cell's job. You're old enough to get out of the house and get a job, Johnny. Come on. I'll help you. Right? The kind of mom that sits at your interview. Okay, some serious stuff here. So regulatory business. Okay. The problem becomes, okay, when helicopter mom becomes a regular oncogene, right? Cancer-causing gene, aka party mom. Okay, that weird mom that definitely should be in jail, throws party in her basement, has like the weird under the stairs liquor cabinet. Okay. It's only midnight, you'll be fine. I've been smoking since I was 11. Right? That mom. Okay. Problem is, that mom is still in charge of the cell, right? So she's telling the cell to divide at inappropriate times. You're little, still divide. Go, go, go. Right? So you've got a bunch of teeny weeny little cells running around. Okay? Cells are active when they should turn off. Okay? So you've got the cell making proteins, right? Think overactive hormones. Okay? Doing stuff when it absolutely should not be doing stuff. Okay? Party mom. All right, that's it. We're going to end there.